trade off. Podium disappeared, but a microphone appeared. Thank you very much. So at least you can hear me. You might not see me in the back row, but at least you can hear me, which might be better. All right. So today we have 2 lectures in the lab, and so we talk about eye and ear. And if you haven't seen it yet, I had released the results for the lecture exam. I don't know if Dr. Choka released the results for the lab exam yet. He did. Okay, I have to look at that. The lecture exam was surprising, let's put it this way. At the upper end, we had really good scores. We had quite a few students who got in the 90%, so 90 to 95% or so. I was a little, well, not a little surprised, really surprised that there were students who got 40 points or 44 points out of 103, and I'm not quite sure how that happens, honestly, because that's why we have review sessions, that's why we have lectures, so you can ask us questions and you can make sure that you know the material. Typically what I do is anyone who got less than 75% should really make an appointment with me, either on the lecture or the lab exam, so we can talk about what we can do, what we can improve, how we can move forward from here. So if that is you, then please send me an email and we're going to meet this week or next week, but certainly before the next exam, or ideally as soon as possible, so we can talk about what can be done to improve your performance. All right, any questions from you at this point? All right, so today we talk about 2 of our sensory systems, the histology of 2 of our sensory systems, starting with the eye. And the eye, as you all know, is a photosensitive organ, and it permits us to analyze the form and color reflected from objects. And the eye can actually self-focus, it can adjust for light intensity, and it can convert light into electrical impulses that are then sent to the brain. So light comes in here at the front. Light comes in here in the front, and then it actually has to move all the way to the back of the eye, where it hits photoreceptor cells in the retina. And the retina actually comprises several layers of cells, including the photoreceptors. You've probably heard about the rods and the cones, and they are located all the way at the back. So light not only has to go through the entire eye, but then it actually has to go through the retina through several layers of cells until it actually hits the photoreceptors. And so this is important to remember because you will see histological images in different orientations, and I want to make sure that you understand where the light actually comes from. So when we talk about the eye, we distinguish a number of structures. So one way to group the structures in the eye is to refer to them as posterior and anterior structures. And the anterior structures are basically the ones here. This is the cornea, the ciliary body here, the ciliary body with the ciliary muscles, then the lens, and here also the limbus. So basically everything here is part of the anterior eye structures, and then the posterior eye structures, basically everything already here. There's some layers here, the so-called sclera, the outermost layer, then the choroid, then there's the retina with the photoreceptors, there's the so-called retinal pigment epithelium. You've heard probably about the blind spot or the optic papilla, and then the fovea. So those are all posterior eye structures. And then here in the in the front part here, in the anterior eye structures, we actually have two chambers. One is referred to as the anterior chamber, this one here, in front of the lens and in front of the iris and the pupil. That's the anterior chamber. And then we have a posterior chamber, which is basically this area here around the lens. And so that's the also referred to as the anterior cavity here, anterior cavity. And then the posterior cavity, sorry, I think I'm going to sneeze anytime soon. The posterior cavity is basically all the white, all the material here with the vitreous body or the vitreous humor, the vitreous body, vitreous humor. So now we're going to talk about in more detail about the different layers here that basically make the eye structure. So there are essentially three coats. There's an external coat or an external layer, that's a fibrous coat, and it has primarily supportive function, and that becomes very important later on when we talk about glaucoma toward the end of the lecture. And it's inelastic, and so in the front, right in the front of the eye, this external layer is in the cornea, and it's transparent, and then it turns into, the cornea becomes the sclera, which is basically when you look at each other, you see that opaque white in your eye, that's the sclera. Then there's a middle layer, which is highly vascularized and pigmented, and that's made up of the choroid further back. So it's melanized, it's brown, rich, lots of vasculature, and it has nutritive function, so it sort of supports the eye. And then closer to the front, we have the ciliary body with ciliary muscles, and they help us to focus the lens. There's some fibers that connect the lens to the ciliary body. And then sort of right in the front is the iris. So that's the pigmented area. That's basically what gives us the eye color, be it blue, brown, green, grayish. And then we have an internal layer, which is basically the retinal coat. And so especially in the back, that is the retina, and the retina is made up of the neuroretina, which is sort of the nervous tissue, with the photoreceptors, the rods and cones, and then some other cell types that we talk about. And then basically in the center of the eye, we have the vitreous body. That's the region behind the lens. It's primarily water, collagen, some hyaluronic acid molecules. So if we now go again to the outermost layer, the sclera, so that's essentially dense, the thick layer of dense irregular connective tissue, and it helps to protect the eye. It maintains the shape of the eye. It's basically this fibrous coat that has supportive function. As I said, it's inelastic, and it's sort of this opaque white. And in the front, then the next layer actually is a layer of loose connective tissue made up, has lots of melanocytes, so it's brown, it absorbs gray light because you have a lot of light getting into the eye, and really the light should hit the photoreceptors, but not all of it hits the photoreceptors, and you want to avoid a situation where the light just strays around. So the choroid is rich in blood vessels, and it has nutritive function. So here's just a histological image. So here would be the center of the eye, right? And the first layer is the retina here, the innermost layer. If you come from the inside of the eye, would be the retina. Then the next layer is the choroid, and you can see it's this melanized layer here. And then outside here is the sclera, dense irregular connective tissue. So again, if you go from the other way, sclera, choroid, retina. And light, depending on where the front of the eye is, light can come anywhere from here or here, depending on where the front of the eye is. Here's another image. So here's the sclera up here, and here's the choroid. So where would the light come from in this picture? So there's basically four options. It's either coming from the top, going down, or it's coming from the bottom, going up, or it's coming from the side. From the bottom, right? Because the sclera is the outermost layer. Here's the choroid. The next layer down here, which we don't see, it's not shown here, would be the retina, and so the light would come from the bottom up, and any stray light then can be absorbed here by the choroid. Okay. All right. So now we are looking at more layers, and especially we are looking at the retina. So again, if you come from the outside, the sclera is the outermost layer, then the next one is the choroid, and then there's basically the several layers of cells which all make up the retina. And the outermost layer is basically a layer of epithelial cells. It's also referred to retinal pigment epithelium, or here it's just called the pigment layer. And then you have the photoreceptors, the rods and cones, and then there's some other cells. So the rods and cones are connected to bipolar cells, and the bipolar cells are connected to these larger ganglion cells. And the ganglion cells then send an axon, because these are all nerve cells, right? The ganglion cells actually send an axon out of the eye to into the brain. And so these are the layers here. Where does the light come from here in this image? Comes from the bottom, right? Because some actually read the arrow here. It says direction of the light. So it comes from the bottom, and so basically the front of the eye would be somewhere down at the very bottom, and then the light has to go all the way through these layers until it actually hits the photoreceptors, and it's specifically the outer, so-called outer segments of the photoreceptors. So let's stick with the retinal pigment epithelium for a moment. So basically this next slide is about this layer here between the choroid and the neuroretina. It's the retinal pigment epithelium. So this is sort of what these cells look like. It's essentially one layer of cells, and they can also absorb light. They have pigment granules in them, and what that does is it prevents the back reflection of light that hits the retina, because once the light hits the retina, it should sort of be absorbed, and it shouldn't go back and re-stimulate the photoreceptors again and again. So it's an epithelium, so there are tight junctions, and so this epithelium actually forms the blood-retinal barrier, because you don't want to have anything get into your eye from the blood, because the choroid, remember, is filled with little blood vessels, and potentially the blood vessels carry some material that could be harmful to the eye or to the photoreceptors. So the retinal pigment epithelium forms this blood-retinal barrier. Now the other thing that it does is, so here you see these so-called outer segments of the photoreceptors. And so the photoreceptors are bombarded by light that comes into our eyes. And these outer segments of the photoreceptors, they're basically looking like stacks of membrane on top of each other, right? So if you look at that, there's basically like stacks of membrane. And it turns out that these outer segments are shedding these membrane layers, or these membrane discs, every night. And it is the retinal pigment epithelial cells that phagocytose these shed membrane discs, and then the photoreceptor makes new ones, and so it's a continuous turnover. So there's this outer segment turnover, and the retinal pigment epithelial cells phagocytose these membrane discs that are shed by the photoreceptors. And you can also see these, I talked about these pigment granules. You can see here, you see all these photoreceptors sticking up there, and between the photoreceptors, the pigment epithelial cells place these pigment granules that can absorb light, so that there's no light getting back onto the photoreceptor. So that's basically what they that's what they do, and so the you can see basically machinery, the metabolic machinery in the pigment epithelial cells that helps to make these pigment granules, and there's also lysosomes that basically digest the membrane fragments that are coming from the photoreceptors. So now if we take a closer look at the photoreceptors, so there's essentially two morphological types, the rods and the cones. And so these photoreceptors, they have essentially an inner segment, and then they have an outer segment here. And the outer segment has these membrane discs here. And there's a cilium in between here. And you remember when there's a cilium, then often you have a lot of mitochondria here. Because what actually happens, and we will learn about that in more detail in the spring, when you learn about the physiology of the eye or of phototransduction. There's a constant ionic current that's going on. That's a so-called dark current. It's essentially a sodium-potassium pump that pumps out ions, and that needs lots of ATP, and therefore you actually have lots of mitochondria here in that in the photoreceptors. Now physiologically, the rods and the cones are also different in that the rods detect low intensity light, so when it's dark in the evening or at night, most of the activity, or when you see something, that's mediated by the rods. So they see images in shades of gray, whereas the cones are there to detect color. They are very sensitive. They help us to discern details of images, and so they work at higher light intensity, so when it's bright out there. And there's three physiological types to detect different wavelengths of light, the blue, green, and red ones. So cone, the way I remember it is cone for color, and then I know that rods must be for shades of gray. Okay, and so there's quite a few of them. So rod photoreceptors, we have about 90 to 120 million per eye. They're basically these, and they are bipolar cells, right? So they essentially have a dendrite cell body, not much of an axon, but they're still considered bipolar cells. Okay, so that's what it looks like. Now, actually, if we go back to the previous slide, here to the retinal pigment epithelium, again we see here the outer segments and the membrane discs that are phagocytosed by the pigment epithelial cells. So, and these cells are more or less cuboidal in shape, as you can see here. And then there's a membrane, or basically like a basement membrane, the so-called Bruch's membrane. So Bruch's membrane, B-R-U-C-H, Bruch's membrane separates the retina from the choroid. The choroid would be here on top, right? And so Bruch's membrane then is composed of the basement membrane of the pigment epithelial cell and the endothelium of some capillaries in the choroid. And the reason I mentioned that is, I'm going to come back to that in a few slides, because there's a pathological condition that you might have heard about, namely detached retina. So we're going to come back to that in a second. So again, here the photoreceptors. Here's a general description of the retina. So the retina has several layers, an outer layer of rods and cones. Then as far as the neuroretina is concerned, so basically the outermost layer would really be the retinal pigment epithelium, and then you have the outer layer of rods and cones. Then you have an intermediate layer of bipolar neurons, and there's actually two other cell types. I don't typically talk much about them. They're referred to as amacrine cells and horizontal cells. I'm going to see that on the next slide. And then there are the ganglion cells that basically carry the information from the eye through the optic papilla to the thalamus. So the retina must be transparent and thin, so you really want to have minimal distortion to not interfere with signal transduction. And these photoreceptors, they actually make graded potentials instead of nerve impulses or action potentials. So now there is no real structural junction between the retinal pigment epithelium and the photoreceptors, and that can actually lead to this phenomenon of the detached retina. So even though it looks like the retinal pigment epithelial cells and the photoreceptors are sort of going at each other like this, there isn't really a structural junction, and that means the photoreceptors, the rods and cones, can actually pull away from the retinal pigment epithelium, and that happens often when there is material accumulating under Bruch's membrane. And so when there's some debris or some dirt coming in from the blood vessels or so, then that can affect the retinal pigment epithelium, and then it actually detaches from the neural retina. So that's referred to as detached retina or retinal detachment. So here's another more colorful image of the retina. Again, up here, this gray line would be Bruch's membrane. Then here's the pigment epithelium. Here the rods and cones. And then you have the bipolar cells. So basically the signal pathways from rods and cones to bipolar cells to the big ganglion cells, and then the ganglion cells project out of the eye. And then you have horizontal cells that are mediating interactions between different photoreceptors, or they help help us to focus or increase the contrast between different input channels. And then the amacrine cells are sort of modulatory cells in the retina. So here's another image, different version of essentially the same thing. Again, up here is the retinal pigment epithelium. Then here you have the photoreceptors. You can sort of see the outer segments of the rods and cones. Here actually the cell bodies and the nuclei and of the photoreceptors. Therefore this is referred to as the outer nuclear layer. Then we have a plexiform layer. A plexiform layer typically just refers to a layer with a lot of processes, a lot of axons and dendrites. So there's an outer plexiform layer. Then there's an inner nuclear layer. That's basically your bipolar cells, horizontal and amacrine cells. So basically we have the outer nuclear layer, the inner nuclear layer, and then we have the ganglion cell layer. And in between here we have another inner plexiform layer. So, so again, photoreceptors, bipolar cells, ganglion cells, and you can see the ganglion cells have been drawn really large here. There's sort of the bigger cells in the retina. Now here's an image, histological image of the wall, including of the eye, including the retina. Again, where would the light come from here? Well, here was the sclera. That's the sclera. That's the choroid. Yeah, so it's from the bottom, right? So again, this would be sclera up here. Here's choroid, melanized, right? Capillaries. Then here you see the single layer of cells. That's the retinal pigment epithelium. Then this would be the outer nuclear layer with the cell bodies of the photoreceptors. And then this is the inner nuclear layer. So those would be bipolar cells, horizontal and amacrine cells. And then here the bigger ones down here, fewer in numbers, are the ganglion cells, or the retinal ganglion cells, and they then send their axon out of the eye through the blind spot or the optic papilla.
What did you say that first layer was called?
This is this or this. This is the outer nuclear layer.
What's in there?
The photoreceptors. So it's basically this layer here with with the cell bodies or the nuclei of the rods and cones.
Okay, thank you.
So outer nuclear layer, inner nuclear layer, ganglion cell layer. So outer nuclear layer, inner nuclear layer, ganglion cell layer. And then in between we call this plexiform layers. And up here, this is basically where we don't really see it, but that's well, we kind of see it. These are the outer segments of the photoreceptors up here. Right? And then the retinal pigment epithelium here. Question?
That layer above, like all the way on the top,
That's the sclera.
the sclera.
Sclera, choroid, and then retina. Yeah. All right. Okay. So we stay at the back portion of the eye, the posterior portion of the eye, to talk about the fovea. The fovea and the macula lutea, and then later on also about the blind spot. So, so again, here's the sclera. This is shown here as essentially just a white layer. Then here, this orange layer, orange brownish, is the choroid, and then in yellow we essentially have the retina here. Okay, and so the fovea is this structure that we find at the posterior pole of the optical axis. And what does that really mean? So if you draw a line right from the center of the eye in the front all the way toward the back, sort of like the direct connection from the front to the back, then you will hit the fovea here. And that is this shallow depression, and it has a very thin retina in the center. And the reason the fovea is so thin is because the bipolar cells and the ganglion cells are basically pushed to the side, to the periphery of this depression, and the only thing that's left in the center are cone cells. And the reason they are cone cells is because cone detects color, right? And the fovea is this area of high acuity vision. So if you want to focus on something, you basically make sure the light goes right down your optical axis until it hits the fovea. And the only cells that are there in the fovea is your cone. So that allows you to really see very clearly what you're focusing on. And in addition, you saw the cones. The cones are typically shaped more like a triangular shape. Well, in the fovea, they actually look more like rods. They're sort of like very, very slender and long, so you can actually put more cones into that area. So that's an adaptation to permit a closer packing of cones and thereby increase the visual acuity. So there's the central region of the fovea, the so-called fovea centralis, or here the central fovea. It's only about 200 micrometers in diameter. And then this fovea centralis is surrounded by the so-called macula lutea. Any idea what macula lutea could translate into? It's the yellow spot, right? The yellow spot. And it's characterized by yellow pigment or xanthophyll pigments. And so basically you have colored pigments there in the inner layers that surround this shallow fovea. And so the macula lutea is produced by these xanthophyll pigments. And what these pigments do, they absorb short wavelength light. And that really helps with our central vision. We don't have any blood vessels there in that particular region. And so why do we need that? Why do we have a macula lutea there that absorbs short wavelength light? Well, short wavelength light, so it's yellow, so the macula lutea is yellow because it absorbs excess blue and ultraviolet light, that enter the eye, and they are very energy rich, and you don't want to damage your photoreceptors. So this macula lutea essentially acts like a natural sunblock. It's almost like sunglasses on your cone photoreceptors, and so that's basically why we have that. So here's an image of the fovea. So this was the image you saw already, right? Again, here's sclera, choroid, and then here's the retina. This is what it looks like for most of the eye, or most of the retina. And then here in the fovea, you can see that here the ganglion cells. There are no ganglion cells, there are no bipolar cells. Basically, all you have here in this region is a high packing density of cone photoreceptors. So it's the area, so the fovea is this area of highest visual acuity. It's the thinnest retinal region, and then again it has a high packing density of cones. All right, so we stay again sort of in the back area, and coming to another clinical manifestation known as age-related macular degeneration. So you've heard now about the macula, and age-related macular degeneration is a major cause of blindness in the Western Hemisphere, and it's caused by loss of photoreceptors in the macula, and this is also related to some debris or some material that might accumulate in Bruch's membrane. So, as I said, the macula lutea is basically like a short wavelength filter, and if there's some damage to the macula lutea, then the basic damage to photoreceptors, and people can't really focus on objects that are right in the center of the visual field. So again, this age-related macular degeneration is correlated with a decrease in these xanthophyll pigments in the macula, presumably because the pigments would normally protect the photoreceptors from light and oxidative damage, but here that is not the case anymore. All right, so that's the fovea and age-related macular degeneration. Then there's this other spot at the back of the eye known as the blind spot or the optic papilla. Now you can imagine that if you think about the eye and the retina, and there's the retinal ganglion cells, and the retinal ganglion cells have to send their axons out of the eye, essentially into the brain. And so that's where this happens. And so basically there is a spot in the back of the eye on the retina where the retina is interrupted because the retinal ganglion cells send their axons as the optic nerve into the brain. And so you can actually try that if you and don't do it now, but everyone I know always does it. If you close one eye and you hold your thumb out there, and you look like, if I would look at the back of the classroom, then if I move my thumb around, at some point the thumb disappears. And the reason the thumb disappears is because the light that reflects from the thumb hits exactly on my blind spot. And so that's what the blind spot is. Because in that portion of the retina, we obviously don't see anything. So anything that reflects light onto that particular spot, we don't see. All right. Oh, okay. So now we are actually moving back to the front of the eye. We're going to talk about the cornea for a second. The cornea is this clear circular area that forms the anterior part of the supporting coat of the eye. It's transparent and it's avascular. It has a convex, not concave, but convex outer surface. It's basically the corneal air interface, and it's really the main site of refraction or light bending. And so what does that mean? So when light hits the cornea, the light rays that might come in as parallel light rays, they are then bent so that they hit exactly on the retina. And ideally, the cornea refracts or bends the light rays so that they really hit the retina, not that they converge on a point before the retina, or they will converge on a theoretical point behind the retina, because then we don't really see well. So now the fact that the cornea is avascular, that means it doesn't have any blood vessels, and that is really important. The cornea here has endothelial cells on the inside. They are also referred to as endothelial cells, and they are unusual in that they don't turn over. So if there's some damage to these cells on the inside, then basically the cornea has a gap there, and if the damage is too big or too large, then you basically create you have some holes here in the cornea, and the cornea then will start to swell up because there's fluid. There's sort of a watery fluid up here. We'll talk about that later. The aqueous humor. That fluid then gets into the cornea and makes the cornea swell up, and it becomes opaque, and that means we can't really see through the cornea. And that is the reason that corneas are transplanted. And they can be transplanted between completely unrelated people because they are avascular. So you can basically take, if someone was in a fatal car accident, you can essentially take the cornea from that person and transplant it onto someone else. And so corneal replacement is probably the most common form of transplant surgery. And again, these transplants can be performed between unrelated individuals because there are no blood vessels to carry white blood cells which would attack the foreign tissue in that specific area. All right, okay, so that's basically what we have here. The other thing that we also have here in the cornea, you've probably experienced that before. The cornea is very sensitive to touch, right? So you don't want to touch it. And that's because there's a lot of free nerve endings reaching into the cornea, which makes it very sensitive to mechanical stimuli or to touching. All right, so here the corneal epithelium continues out here, and this little line here, that's actually the conjunctiva that's coming out of here. It's a stratified or columnar epithelium, secretes some fluids or some mucus out there. All right, so here's another diagrammatic version of the cornea. Again, here's the epithelium on the outside. Then there's, and it's a stratified, non-keratinized squamous epithelium, as you can probably detect yourself. You can see there's some nerve fibers going in here, some free nerve endings. And then there's this thick basement membrane known as Bowman's membrane here. And then the center of the cornea is referred to as the stroma or the substantia propria or stroma. That's the center portion of the cornea, and it basically makes up most of the cornea. And that is another example where we actually have dense regular connective tissue. We talk about dense regular connective tissue when we talk about tendons and ligaments. But here's another example of a dense regular connective tissue. And so the fibroblasts here and the cells they're very well arranged, very well organized, so that light can pass through without any major problems. So here's another image, actually a histological image. Here's the epithelium. Here's the basement membrane under it. It's kind of difficult to see. Then here's the stroma of the cornea, or the substantia propria. And then here on the others, on the sort of the side that faces the eye, we have this endothelium. And as I said, the endothelium, that's an endothelium where the cells they're not regenerated. So if something happens to these cells, then you get gaps in the endothelium, and then the fluid that you find here can get into the cornea and basically make the cornea swell up, and the cornea becomes opaque. Now this endothelium also has a basement membrane under it. That's referred to as Descemet's membrane. So Descemet's membrane is this basement membrane under the endothelium, and if you look carefully, you can see this pink line here. That's Descemet's membrane here. And there, this pink line, especially up here, you can see it. Now, the corneal endothelium is special in the sense that it actually transports ions across the membrane. And so, as I said, the stroma here of the cornea needs to be in a somewhat dehydrated state, because if there's water in there, then it becomes opaque and the light doesn't really go through. So there shouldn't be any water in the cornea. And so what these corneal endothelial cells do, they basically pump ions from the cornea into this area. This is actually the anterior chamber. So here, this is the anterior chamber. And the endothelial cells pump ions from the cornea, from the stroma, into the anterior chamber. And these ions are mostly bicarbonate ions. And when they do that, so what happens if you pump ion from here to there? Well, if there's any water here, then the water would follow osmotically in this direction, and what that does is it basically keeps the stroma dehydrated. And that's what we need to do, because if this is all opaque, then that would be a candidate for a corneal transplant. So that's basically what it is, and you probably all have heard, or maybe even some of you have done that. You have heard about LASIK surgery, right? That's essentially what it does. It's a sort of a kind of laser surgery, and it's LASIK stands for laser assisted in situ keratomyolysis. So don't write that down. And so, if you have any issues with your cornea, either it's swollen or it doesn't have the right shape. As I said, the cornea is there to bend the light rays so that the light rays hit the retina, and if it doesn't hit the retina, then the surgeon can go in there and basically take portions of the cornea out to make sure that the cornea is shaped such that the light rays hit the retina. So as I said, the cornea, this is an image of the stroma of the cornea, and you can see this packing of the collagen fibers here. The packing, there's all these collagen fibers. There's a very regular arrangement of these collagen fibers, and they're either going in one direction or at a 90-degree angle. So there's not, they don't go any which way, and that means that there's little diffraction of the light. So that light goes right through there without any problems. All right, next point is the iris. The iris separates, so the iris is here, this portion here in the front, and if you look at diagrams, of course it looks like there's an iris coming from the top and the bottom, which of course is not true. These are all circular structures, right? So the iris is basically this circular structure, and this hole that the iris forms, we refer to that as a pupil. But the pupil is not an anatomical structure. The pupil is essentially the hole that's created by the iris. And when the iris opens up, then the pupil becomes bigger, and when the muscles in the iris contract, then the pupil becomes smaller, right? So here's the iris, separates anterior and posterior chamber, and it gives us the eye color. So basically, there are melanin-containing cells, again, in the stroma, in the center of the iris, and depending on the concentration and on the distribution and on the cellular density and the scattering of the light, we all have different eye colors. But as a rule, if someone has lots of melanocytes, then that person would have brown eyes, and if someone else has relatively few melanocytes, then that person would have blue eyes. So that's essentially how light color comes about. And here is another diagram. Again, here the so-called posterior, the anterior chamber and the posterior chamber. Here was the iris, the cornea. Here's the iris out here. And then basically here's the cornea, which then would become the iris over here. This structure that's taken off here is the conjunctiva.